Okay, today we'll be looking at the properties of DNA replication. Replication is a self-duplication process in which a molecule acts as a template and produces its own copies. So when you have DNA replication, DNA is used as a template and it produces a DNA copy. So that's why DNA replication is DNA directed DNA polymerization. When you look at the characteristics, the first character that we consider, which is almost universal, is the replication process is semi-conservative. And when you say semi-conservative, 50% of the parental DNA will be conserved. So whenever a new DNA molecule is synthesized, 50% of that will be parental one, 50% of that will be newly synthesized. So if this is the parental one, parental DNA, when you have semi-conservative mechanism, each daughter molecule gets one parental strand and synthesizes the complementary nucleotides. So these are the daughter DNA molecules. Or you can say DNA copies. In that, this is parental, those two parental ones. This is newly synthesized, this is newly synthesized. This particular feature of replication was given by Watson and Crick. This was published in their Nature newsletter of April 1953. And the reason why they thought of this kind of replication, where one strand will be acting as a template, one strand will be synthesized, is the two strands of DNA are complementary. And not identical. So this is the important feature of the double helix molecule. So one of the features they said in the double helix molecules is the two strands of the DNA are not identical, but they are complementary. The nucleotide sequence in one strand determines the nucleotide sequence in the other strand. So this particular feature is useful in our semi-conservative replication because the nucleotide sequence of the parental DNA strand determines the nucleotide sequence of the newly synthesized strand, so that the newly synthesized strand will have complementary nucleotides. So that is one of the easy ways you can go on synthesizing the DNA through the self-duplication process. The experimental evidence for this mechanism was provided by Michelson and Stahl. So they used E. coli as the organism They use two different nitrogen isotopes. One, the light isotope, what we call it as N14. The second one, the heavy isotope, what we call it as 15N. And they had ammonium chlorides of light and heavy nitrogen. These ammonium chlorides are provided as a nitrogen source, and then they carried out the experiment. First, they carried out the growth for several days in N15 medium. When it was grown for several days in N15 medium, every nitrogen atom present in the nucleotide became N15, it became heavy. After that, they transferred the cells to the N14 medium. Once they transferred the cells into the N14 medium, they waited for 40 minutes, the generation time of E. coli. After every 40 minutes, they isolated the bacterial cells, extracted the DNA, subjected to the DNA to isopycnic density gradient centrifugation or equilibrium density gradient centrifugation. Now this is an experiment where the molecules will be separated by bond density. So molecular separation is based on bond density, or usually we write the Greek letter rho. So if two DNA molecules have differences in their bond density, they will get separated, they will form distinct bands in the case of equilibrium density gradient centrifugation. Since N14 and N15 have different densities, they are going to produce two distinct bands. So when they carried out this experiment, what they found after the first generation is they got only hybrid DNA. So that is after the first generation. 100% hybrid DNA molecules having N15 and N14 strands. When they have carried out the isopycnic centrifugation, they got an intermediate band. That intermediate band indicates hybrid DNA. So what we can conclude from this experiment? Since we are getting only hybrid DNA, we are not getting light DNA, we are not getting heavy DNA, we can say conservative replication is not possible. Replication cannot be conservative. If it is conservative, then we should have got two bands, one corresponding to heavy DNA, one corresponding to the lighter DNA. Since we are not getting that kind of it, you can prove that replication is not conservative. Then when they went for the second generation, what they found is, in addition to the hybrid DNA, they also had a lighter DNA. So now how we can explain this DNA strand from this this DNA strand from this DNA strand from this DNA strand from this. So this is N15, N14 hybrid DNA. This is N15, N14 hybrid DNA. This is N14, N14 lighter DNA. This is N14, N14 lighter DNA. So we got 50% of hybrid DNA and 50% of lighter DNA. That experimentally proves replication is taking place by semi-conservative mechanism. So after 2 generations, if you go for the isopycnic density gradient centrifugation, you get one intermediate band. So that is N15, N14, and you get one lighter DNA. Where both the strands are N. And the gradient here, whatever you have, this is cesium chloride gradient. So for DNA experiments, the preferred gradient is cesium chloride gradient because it gives relatively high densities. So you can get 1.8, 1.9, 2, 2.1 and so on. And this high density is needed because DNA is a highly viscous, high buoyant density molecule. So that's about the first character. Replication is semi-conservative. When the new daughter molecule is formed, one strand will be that of parental strand, another strand will be that of newly synthesized strand, and therefore always 50% of the parental DNA will be conserved in the daughter DNA molecules. The second property of replication is semi-discontinuous. That means half continuous, half discontinuous with respect to the two newly synthesized DNA. So, one newly synthesized DNA strand will be continuously synthesized. Whereas the second newly synthesized strand will be discontinuously synthesized. Since half is continuously synthesized and half is discontinuously synthesized, we call the process as semi-discontinuous. The strand which is continuously synthesized is called leading strand. And usually requires a single primer. So the continuously synthesized strand is called leading strand. Leading strand synthesis starts with a single primer, and that primer will be continuously extended. The one which is discontinuously synthesized is called lagging strand. The lagging strand will be synthesized in bits and pieces. So it's not like one piece, but several pieces are synthesized. These bits and pieces which are formed during the lagging strand are called Okazaki fragments. Every Okazaki fragment starts with RNA primer. And is extended by DNA. So if you just draw a diagram, this, let us say, the primer, that is RNA, and the primer is extended. This is the DNA part. And then you have mechanisms where the primer RNA will be removed and then it is replaced with deoxyribonucleotides so that the entire lagging strand will become DNA nucleotides. The Okazaki fragments require another enzyme called DNA ligase. This DNA ligase joins the Okazaki fragments. So if you have the DNA ligase, you will see the continued lagging strand. If you don't have the DNA ligase, then Okazaki fragments remain as such. So your lagging strand will be synthesized in the form of small pieces. Then when you look at the diagram in terms of all these features, we will take one side of the replication, the other side also exactly the same thing happens. So I am drawing one half of the replication. So this is one strand, template strand. This is another template strand. This strand, let us say orientation is 3'. This is 5'. In that case, this should be complementary, 5'. It should be 3'. So these are template strands. So we call this as leading strand template. This we call it as lagging strand template. The place where actually the replication begins is called origin. So the DNA strand which is oriented 3' to 5' with respect to the RE is the leading strand. The DNA strand which is oriented 5' to 3' with respect to the RE is the lagging strand template. RE stands for origin of replication. So the first region that undergoes melting, localized separation and begins the synthesis of replication is called RE. In E. coli, this is specifically called as REC. So first we have a primer. This is the primer. The primer will be 5', 3'. This strand is 3', 5', so it is complementary. Then the polymerase extends this RNA with the deoxynucleotides. So there you have the polymerase. So this will become the leading strand. This is RNA primer. When it comes to the other strand, you cannot synthesize this direction because DNA synthesis is always 5' to 3'. It cannot occur 3' to 5'. So this is fine 5' to 3'. But if I put a primer here, it will become 5' to 3'. There is nothing to extend. So what happens with respect to the lagging strand is the primers are formed in the inner side. So that is the primer. This primer is 5', 3'. And that primer will be extended by the DNA polymerase, generating Okazaki fragment. Then, as the leading strand is further synthesized, one more primer is formed, and this primer is further extended. So you have formation of the primer and then extension of the primer by deoxynucleotides. That's how the Okazaki fragments are synthesized. So this is one more Okazaki fragment. Then somewhere here we get one more primer, and the polymerase starts synthesizing DNA. So this is the polymerase. So core polymerase, and these two are joined by a dimerization unit. This we call it as the tau unit. So the function of the tau unit, the dimerization unit, is to ensure that leading and lagging strands are synthesized to the same extent. You don't have a case where leading strand simply runs off and the lagging strand will be delayed and waiting for completion. So you don't have that kind of process when you dimerize both leading strand polymerase and lagging strand polymerase. Even though in this diagram it appears that they are moving in the opposite side, usually we have the formation of a loop structure. That loop structure makes both the polymerases to move in the same direction as dimers. Now, in order to describe the leading strand based on the diagram, and also the way you can identify if a diagram is given. Leading strand is something that begins at origin. It moves away from origin. It moves in the direction of that Y-shaped structure. The Y-shaped structure which is formed during the replication is called replication fork. And this is the direction in which the fork is moving. So, leading strand moves away from the origin into the direction of replication fork. So basically, it is chasing replication fork. So leading strand chases replication fork. Now when you look at the lagging strand, the things are different. So lagging strand is synthesized in this direction. So it is moving away from the replication fork. It is moving in the direction of origin. So moves away from replication fork into the direction of origin. So this is the easiest way you can identify whether a given DNA molecule strand is leading or lagging. Leading moves away from the origin, lagging moves towards the origin. Leading moves towards the replication fork, lagging moves away from the replication fork. Here we will have the enzyme that will be making unwinding of DNA. So this enzyme we call it as DNA helicase. The DNA helicase is an enzyme that unwinds the DNA so that the replication fork can progress forward direction. Here we have another enzyme which is called topoisomerase, or bacteria we call it as gyrase. This enzyme actually decreases the positive supercoils which are generated as a result of unwinding of the DNA. So if the topoisomerase works, then DNA helicase works. DNA helicase is ATP dependent enzyme. It requires one ATP hydrolysis for every one turn it removes. And then once your helicase has done the unwinding, you get more amount of replication fork. Your leading strand synthesis extends. Once the leading strand synthesis is extended, you have a new primer formed for the lagging strand, and that primer will be extended by its own polymerase. So this is the second feature, semi-discontinuous mechanism. The third property is bidirectional replication. So when I discussed about the semi-discontinuous mechanism, I said I am showing one half of the replication fork. I have not shown both the sides of replication fork. In a bidirectional replication, if this is the position, what we call it as origin, then the helicases, one helicase bound to this, one helicase bound to this, will unwind the DNA so that we get two Y-shaped structures moving in the opposite direction. So that is origin. So this fork is moving in this direction, this fork is moving in this direction. Replication fork, replication fork. Now we will have at least 4 polymerases associated with this. This will be one leading strand. This will be another leading strand. These are the lagging strands. So we have 3 prime, 5 prime, 5 prime, 3 prime. So this is leading strand for this template. This is lagging strand for this template. This is leading strand for this template, and this is the lagging strand for this template. So once you know the origin, you know the directionality of the 2 strands, it is always possible for you to identify correctly which is leading strand, which is lagging strand. So the replication moves in this direction with the same speed as it moves in this direction. And as a result, because of the bidirectional replication, the time required for replication is decreased. Now this easily you can understand by taking E. coli. If it is unidirectional replication, where replication moves only in one direction, it requires about 90 minutes. That's 1.5 hours you need for the complete replication of the 4.6 million base pairs. In the same E. coli, under normal condition where the replication is bidirectional, you need about 40 minutes. So almost a 50% reduction in terms of the time takes place because of the bidirectional replication. So in case of a long linear chromosomes, when you have something like this, you see several regions where replication is originated and forming replication bubbles in bidirection. So each one has an origin that is initiated. From each origin, the replication is moving in both the directions. This is with respect to the eukaryotic linear chromosomes. When you take bacteria, remember they have circular chromosomes. So somewhere here we have RE region, almost 180 degrees opposite to that we will have termination region. This is how the replication DNA occurs under the microscope, electron microscope. So this is the leading strand. This is the leading strand. This is the lagging strand. This is the lagging strand. So when you observe under the microscope, it appears this way. That is in the theta shape. So Keynes, when first he studied, he gave theta model of replication. So it was thought as a new replication method. Later on, people identified that the theta mode is not at all a new type, it is bidirectional mechanism. So terminations, you will have one termination here, almost 180 degrees. So that we call it as ter-eta. And we have the second termination this side, which we call it as ter-BC. So these ter regions, they will bind TUS proteins and they stop polymerase. So that's in case of the bidirectional replication. The next character that we can consider is the replicons and origins of replication. So whatever the amount of DNA that you have, if that entire amount of DNA, for example in this case, if this entire amount of the DNA undergoes replication as a single unit, then we call that as replicon. So the entire unit of DNA that undergoes replication as a single unit. So a replicon is the unit of replication. Every replicon contains a point where the replication is initiated. And this point we call it as an origin. So replicon is the basic unit of replication. Origin is a position in the replicon where the replication process begins, the initiation process begins, initiation protein binds, and so on. All that initial changes that you require in terms of the activity you will see at the origin. When you look at bacteria, they have a single replicon. So in bacteria, replicon size will be equal to genome size. So if you take E. coli, genome size is 4.6 million base pairs, and because of that, the replicon size is also 4.6 into 10 power 6 base. So bacteria, key things to remember is one origin, one replicon, two terminus points, another important thing when it comes to the bacteria is when replication is initiated, it is initiated always at one position because there is only one origin. So replication in bacterial genome is initiated from a single point, as there is only one origin, it occurs from only one point. But the advantage the bacterium have is an origin in bacterium or in bacteria can be activated more than once. So we say can be fired more than one time. This is something that never occurs in the case of eukaryotes. So you can start the replication and the replication reaches this point. Again you can have another round of replication, and even you may have a third round of replication. So an origin can be used multiple times in bacteria. Bacteria have only one origin. Bacterial replicon is equal to bacterial genomic size. The entire genome can be subjected to replications multiple times at a single position. And this is very important when the bacterial cells have generation times that are very small compared to that of the usual generation time. For example, in the case of E. coli, the usual generation time that you need is 1 hour. But however, if you provide optimal conditions, the bacterium undergoes generation time of 30 minutes. So that means it is able to replicate a part of its genome in the previous generation, and therefore the amount of the genome that has to be replicated in this generation is significantly decreased. What about the eukaryotes? When you take eukaryotes, you have distinctions. The first distinction is bacteria generally, the entire genome is organized into one chromosome. Eukaryotes, genome is usually organized into more than one chromosome. For example, in the case of Drosophila, we have 4 chromosomes in the genome. In the case of humans, we have 23 chromosomes in the genome. Pisum sativum, we have 7 chromosomes in the genome. So the genome is organized into more than one chromosome. Next point, each chromosome is divided into several thousands of replicons. Now, how many replicons you have depends upon size of the genome, size of the chromosome, and size of the replicon. All this will determine how many replicons are present in a given eukaryotic system. Each replicon varies in size from 25 kilobase pairs in primitive lower eukaryotes to 300 kilobase pairs to vertebrates. So a human genome contains replicon size of about 300 kilobase pairs. Now see that these numbers are far small when you compare with that of the bacterial case, where a replicon is 10 power 6 base pairs. So bacterial replicons are larger than eukaryotic replicons. Then, like the bacterial case, each replicon has its own origin. So one replicon, one origin, that is true everywhere. So what happens here is, so let us just make a boundary that each bounded one is a replicon. Now, each one has an origin. So each one is a replicon. You may have a replicon activated here, the origin activated here, 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 origin activated here. So it's not that in the case of eukaryotes, all the origins have to be fired at the same time. Some origins fire, at least 10% of the origins will not fire in a given cell cycle. This is a sort of a defense mechanism to make sure that if any replicon fails to undergo the replication, the adjacent replicon can complete the process of replication. So as a result, when you look at the bacteria, the eukaryotic chromosome under microscope, electron microscope, you see replication bubbles formed at several positions on the chromosome, unlike E. coli, where your replication bubble is found only at one single position. So eukaryotic origin can be used multiple times. Bacterial origin can be used multiple times, whereas eukaryotic origin can be used only once. In eukaryotes, each chromosome contains several thousands of replicons. Not every origin is used during the given replication. Some origins will not be fired. They will be fired only when you require a condition where firing becomes essential to complete the replication of adjacent replicon. So these are the fundamental distinctions between the bacterial case and eukaryotic case. The next character is primer and template dependent process. And you can always remember as a rule, when we say primer requirement is a must for DNA synthesis. So it doesn't matter how you synthesize the DNA. You may synthesize by using DNA polymerase. You may synthesize by using reverse transcriptase. But the characteristic feature is you need a primer. A primer is an oligonucleotide segment, maybe 10 nucleotides, 50 nucleotides, 20 nucleotides, 30 nucleotides, that contains a free hydroxyl group at the 3' end. So irrespective of how many nucleotides you have here, the important thing for the primer is it should have a free hydroxyl group, because to this free hydroxyl group, the next nucleotide is added by phosphodiester bond. And again it produces a free hydroxyl group. Again another nucleotide will be added. Again through phosphodiester bond. So as long as you generate priming end and you have a template, you can go on synthesizing the DNA. So primer requirement is unique for DNA synthesis. The primers that are synthesized in vivo with respect to the genomic DNA replication are ribonucleotide primers. These ribonucleotide primers will be later on replaced with deoxynucleotides. So once the replication process is completed, we will see only deoxynucleotides. We don't see any ribonucleotide. In bacteria, we have a DNA primase. This enzyme synthesizes primers for both leading and lagging strand. In eukaryotes, there is no distinct primase. So you don't have a separate primase enzyme in eukaryotes. The primase activity is taken by DNA polymerase alpha. So it contains primase activity, and it is the one that synthesizes the primers for both leading and the lagging strands. Then when you take the template, it is the molecule that provides nucleotide information, and based on this nucleotide information, you have incorporation of complementary nucleotides. Into the new DNA strand. So it is a template that provides which nucleotide has to be incorporated at any given position when the DNA is extended. The incoming nucleotide should have ability to pair with the template nucleotide. If the pairing takes place, then you have the possibility of hydrolysis of pyrophosphate and then phosphodiester bond formation. If you have no strong pairing, then it will try with another nucleotide. So the polymerization process, whether it is in the case of the DNA replication or in the case of transcription, is a trial and error process where you try with a nucleotide. If it is able to form stable bond, then you allow its incorporation. If you try with a nucleotide that is not able to form stable base pairing, then this will not be subjected to hydrolysis, and as a result, typically that nucleotide is released from the cleft region of the polymerase. So this actually makes your process of DNA replication relatively accurate, because you are not randomly putting the nucleotides, you are putting the nucleotides based on the complementarity to the template